Welcome to my channel Adhyan with Flow. Let's start with our new topic, fermentation. This process will occur in the absence of oxygen. Okay, in fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic condition by sets of reaction where pyruvic acid is converted to CO2 and ethanol. We can betterly understand by this chart. Let's start. This is the glycolysis pathway. Glycolysis pathway, here to here. We know the end product of glycolysis is pyruvate pyruvic acid. Here, this is the end product of glycolysis. Glycolysis is a 10-step process. We know now. We were study before. We already studied this. This glycolysis is 10-step process. The next step, this is the end product of glycolysis. We found 2 molecule of pyruvic acid, and if we provide oxygen to pyruvic acid, they goes into TCA cycle. But if we do not provide oxygen, then they goes into 2 pathway. They form lactic acid and they also form alcohol or ethanol plus CO2. I repeat again, the end product of glycolysis, we found 2 molecule of pyruvic acid. We found pyruvic acid, and in the absence of oxygen, we get lactic acid in muscles, and we get ethanol and CO2 by yeast. They form this process, and our muscles form this process. If we doing some muscular exercise, that time lactic acid produced in our body. So muscle lack, they need oxygen. During of less fulfillment of oxygen, lactic acid is produced, and pyruvic acid to lactic acid conversion, we reduce some energy. Here also, pyruvic acid to ethanol, we lose some energy, like NADH form NAD plus, and NADH here is again we lost some energy and we get NADH. So here is loss of energy, but here we get energy. So there are compensation. So there are compensation of energy. Clear, guys? Very clear. So this is the master table. If you remember this table, you can elaborate fermentation process easily. Means glycolysis pathway. Okay, the end product is pyruvic acid. This pyruvic acid, if they have oxygen, so they can go in TCA cycle. If they do not have any oxygen, they go towards lactic acid. If they do not have oxygen level, in the case of yeast, they produce alcohol. We can call it as a ethanol. They are useful in industry level, plus CO2. This is the fermentation, guys. This is the fermentation. Important. If you understand this chart, you can easily write all the things. You should explain first glycolysis, then show, then how pyruvic acid will form, and pyruvic acid how they convert into lactic acid and ethanol. So this is the one of the example of anaerobic respiration. So that's all about fermentation, guys. If you understand this chart, you can solve any kind of questions. But you should remember glycolysis pathway. You should explain in your exam briefly. You have to remember this 10 step, guys. You have to remember, guys. Let's start with some aerobic respiration. Before starting, you have to know how this reaction will form. We know now, pyruvic acid come from the glycolysis product. At the ending of glycolysis, I tell you now, pyruvic acid will form. If they get oxygen, then they go toward the TCA cycle. If they do not get oxygen, so they produce lactic acid and ethanol plus CO2. So here we provide oxygen to pyruvic acid. Now this mechanism will form pyruvic acid plus coenzyme A and coenzyme A and NAD plus. This is also energy form, guys. But why we doing this thing? Because during this reaction, they goes into TCA cycle and TCA gives us energy. That's why we're doing this thing. So pyruvic acid goes into this reaction. some magnesium ions will require, and yes, most important enzyme, pyruvate dehydrogenase. Important guys, this is the important enzyme. Many time in examination they asked which kind of they give some options, and one of the option is this, pyruvate dehydrogenase. This is the option. Okay, which kind of enzyme convert pyruvic acid into acetyl coenzyme? That time you have to remember pyruvate dehydrogenase, guys. You have to remember multiple time this question will asked, because in the flow of the reactions sometimes we forget to remember this reaction. Then how acetyl coenzyme enter in Krebs cycle and how this acetyl coenzyme will form. This reaction is very important because due to combination of pyruvic acid coenzyme NADA plus some enzymes and magnesium ion, we get what? Acetyl coenzyme, some CO2, we can remove this, and energy also, guys. In this reaction we get energy. So how you can skip this? How? Now let's start. This is the important. Now this acetyl coenzyme goes into where guys? Your favorite TCA cycle. What TCA cycle? We can call it as a oxaloacetic acid. Yeah, this is the name TCA, as well as we can call it as a oxaloacetic acid, OAA, because this is the first product of this cycle. This reaction is catalyzed by the enzyme citrate synthase. Remember this enzyme. Again, I'm repeating, remember this enzyme. You have to remember all the enzyme if you want to clear your concepts about metabolism. You have to clear glycolysis, you have to clear fermentation, you have to clear TCA, you have to clear ETC transport. You have to clear if you want to score full out of full marks in metabolism. And other than C4 cycle, C3 cycle, C2 cycle, you should remember this cycle if you want to perform. If you do not want lose any marks from this, you have to remember. And this is easy, guys. Written practice. Your brain is so smart. So practice. The reaction is catalyzed by enzyme citrate synthase. Synthase means here no ATP is required. If I use synthetase, means ATP is required. If I add on extra T, it means ATP required. If I don't use extra T, guys, T, normal term. If we use extra T, it means they require ATP. This enzyme do not require this kind of molecule. So the reaction is catalyzed by enzyme citrate synthase. I repeat at least 3 times, guys. And a molecule of coenzyme is released. After this enzyme, coenzyme will release pyruvic acid, coenzyme A and NAD. They release after some time. They're helpful in reaction. Citrate is then isomerized to isocitrate. Now, due to this enzyme, coenzyme is released and citrate convert into isocitrate. Citrate is converged into isocitrate. What they lose? Coenzyme A. And what kind of enzyme is present? Citrate synthase. This reaction followed by 2 successive steps. Decarboxylation, leading the formation of first step is formation of alpha ketoglutaric acid, and then look at here, succinyl coenzyme A. And now remaining step is, this is the step 1, and step 2 is now succinyl coenzyme A, they oxidize into OAA, means citrate convert into alpha ketoglutaric and they convert into succinyl. This is the first step. And another state is conversion of succinyl to OAA is the second step B. Look at here the cycle. Let's begin with the cycle. Pyruvate, this is the product of glycolysis pathway. The end product of glycolysis pathway is glucose finally converts into pyruvate. If pyruvate they got oxygen, then they go towards the TCA cycle. If pyruvate does not get any kind of oxygen, so they goes into lactic acid pathway and ethanol fermentation pathway. So this is this pathway because we provide oxygen. So conversion of pyruvate to acetyl coenzyme A, we studied previously. Look at here. Already we studied this important reaction. Important, guys. Never forget this reaction. Pyruvate to acetyl coenzyme. How they form with the presence of this enzyme. Never forget. So pyruvate converts into acetyl coenzyme. We require what? NAD plus and coenzyme. That's it. And NAD plus converts into this energy form. So we get energy here. Now CO2 will also remove. Look at here, guys. CO2 is also removed, energy is also removed, and acetyl is the co-product. So yeah, this reaction is perfect. We got the same thing. Yeah, this is true, very true now. Now acetyl coenzyme, this is the 2 carbon product. Why 2 carbon product? Pyruvate acid is a 3 carbon product. Guys, look at here, there is 1 carbon product is removed now. So remaining is 2 carbon product, and now they make aggregate multiple acetyl coenzyme. They form citric acid 2 plus 2 plus 2. Come on guys, 6 carbon product, citric acid. Now citric acid change into alpha glutaric acid. This process is decarboxylation. No, step 1, citrate to alpha keto, step 1, alpha ketosuccinyl, step 1. So we get another CO2 and we get another energy molecule, energy. So after we get alpha ketoglutaric acid, we now finally have 5 carbon. Why? Because 1 carbon is removed here. So after 6 carbon we get 5 carbon compound, and again we lost 1 carbon and we get energy. Again we get energy. So this alpha ketoglutaric acid, they converts into succinic acid, 4 carbon compound. Because we lost now 1 carbon, we get energy, and also get energy from the GTP form. Guys, ATP, GTP, CTP, all are the energy form. This is the most ancient form of energy form. This is also energy form. NADH also energy form. FADH also energy form. Guys, relax, don't be panic. Now this is completion of step 1. Now here is step 2. Succinic acid, 4 carbon compound change into malic acid, and they give us what? Energy. Energy means TCA cycle provide us highest energy, guys. Now malic acid change into oxalic acid, OAA. Now further they give us energy. Your NCERT, what they do? They give only important step. Like those steps we get energy, they write down here. This is the multiple step, guys. In citric acid they contain multiple steps, but your book remove this step because that step we don't find any kind of energy. That's why they remove. So this is the energy form. Dekho, look at here, energy, wait a minute, energy, and here also energy. So let's calculate. We know now we have pyruvic acid. Look at here, this is the pyruvic acid. Look at here, up pyruvic acid. Now we use some 4 NAD plus. Let's count now, guys. Let's count. 1 NADA plus, second NADA plus, third NADA plus, and 4 NADA plus. Yeah, we use FAD plus. Yes, we use FAD plus. Yes, we use. Yes, we also use some water molecules, but these steps not present here. Here we got water molecule, some ADP and PI. We can also write GDP plus PI. Don't be panic, guys. And this TCA cycle, where is occur? Mitochondrial matrix. Look at here, guys. This is the mitochondria outer membrane. This is the inner membrane of mitochondria. This is the cristae, F node, F1 particle. There is F1 particle, and this is the F node. Yeah, this is the matrix part. Here this phenomena occur. Now we got 3 CO2. Let's count now. 3 CO2. One is here, and one is here. Yes, 3 CO2. 4 NAD plus, they give 4 NADH. Yeah, 4 NADA plus molecule. Of course, they give 4 NADA plus energy. And now some hydrogen ions, they stay in the lumen of mitochondria. Another energy, FADH, 1 FAD plus form 1 FADH2, this energy. And now ATP. Yes, here we got ATP. So in this Krebs cycle, TCA cycle, oxaloacetic cycle, citric cycle, we got highest energy here. We got highest energy. If we break NAD plus, if we break NADH, this energy form, they gives 2 to 3 ATP molecule. If we break FADH, so they also give 2 to 3 molecule of ATP, and this is the simplest form of energy. They also give ATP. Yeah, 3 hydrogen ions combined, and they also give ATP, guys. With the help of ATP synthesis pump, they goes into ETC pathway. So highest energy we will get from this cycle, TCA cycle, and most highest energy we will found in ETC, guys. They gives highest energy. Now you understand, guys, where we start. We start with a simple definition like respiration. What we do? We break our food. we break our lovely fruit and lost some oxygen, and we get what? CO2, H2O, and little bit energy. Why we get food, guys? Why we get food? So we can get energy. And now this food, they give us to energy with the form of glycolysis pathway, with the form of TCA cycle, with the form of ET. This ATP provided by this pathway, this pathway, this pathway. Okay, so food gives us to energy in this form. They produce this pathway, this pathway, this pathway. They produce ATP by this pathway, this pathway, this pathway. Glycolysis we covered in our previous lecture. TCA cycle recently we covered, and in our next video we will finish our ETC. We can also set ETS, guys. Don't be panic. Okay, look at here. Okay, ETS, we can call it as a ETS also, huh? Don't be panic. Now we get this energy molecule, and this hydrogen ion. This molecules goes into electron transport system. We want to break we want to break this thing. I already told you that complex molecule, if we want to change into the simple molecule, they proceed in multiple steps. This is not a one-way step, guys. Complex molecule not easily break down into simplest molecule. They require multiple steps, guys. Multiple. Nothing is easy. Okay, nothing is easy. Everything is inside in our body. Don't worry. So this is ETS. Look at here. What a beautiful diagram here. We will discuss in our next video. So that's all for now. So if still you don't subscribe my channel Adhyan with Flow, then subscribe, guys. It supports me, it encourages me, and keep smiling. Thank you.